Now or moving forward for the rest of the day that you do take note of the time that you're scheduled for. And then reference Facebook Live this morning. So we had 1,678 views, and we've reached 3,493. And then just as a reference for last year when we did Facebook Live, take note of the time that you're scheduled for. And then reference Facebook Live this morning.
We've had a big year in the lab, and it was exactly a year ago today that we received the 55 boxes of remains, and we showed you pictures last year of what those look like. And we've been very, very busy since the very day they came into the lab. And Jen's team has worked very hard on that, as well as on disinterments. And we're very pleased to announce, as you've heard already, that we've already made 8 IDs as of this moment today. But when we talk about there's going to be 25 more is what we've been saying this week. Those are the ones we already know about. Those are the ones we're doing the final reporting on, getting all the reports together. And then there are going to be more after that. And I want to say that the reason we're having all of this success now, almost exactly a year later, is because of some really outstanding work done by Dr. McMahon's lab. The AFME's DNA lab at Dover. And he's going to get up in a few minutes and talk about all the great work they're doing, but I'm going to tell you what he's not going to tell you. That's a great lab. There is no other lab in the world that can do what they do and do it as well as they do with the success rates that they've had. So I want to say that we've had a lot of success, but it's been due to the efforts of a lot of people. And that laboratory deserves a big heaping helping of the credit. And I would tell you, if it weren't for the quality of the work they do, it would not happen like this. We might have 1 or 2 IDs and struggle with the rest, but we're doing extremely well, and there's a lot of success yet to come because of the high quality work that they're doing and what's happening in our laboratory with Jenny Jen's team. That said, this year, in fact, several weeks ago, we hit a milestone for our laboratory. We made the 500th Korean War ID. And this year, we're on track to set a new record for the number of Korean War IDs made in a single year by our laboratory. So we're very pleased at the progress, and we would just tell you to watch that a lot of good things are yet to come. Now, from the laboratory here, we have Dr. Veronica Keys over here on the side. Veronica's going to give you a presentation tomorrow on the K55 project and then other things that are being done by the Korean War Identification Project. Dr. Jen couldn't be here this year. You may remember last year, if you talked to her, that she was quite pregnant. And that was a big success. She has a beautiful baby daughter, and she's trying to take some time off this summer to spend quality time with her baby. And so she wasn't able to make the trip, but her second in command is Dr. Keys. She also went into North Korea with us last summer, and she's going to give you an update on the Korean War Identification Project tomorrow, and she's happy to talk to you, answer questions about what's going on, and she's going to be wonderful. Way back in the back corner is Dr. Greg Byrd. He's one of our managers from the lab. And he's here this year. He's never been here before. He was whining and moping about a month ago, saying, how come I never get to go to the Korea War Conference? So this year we brought Greg, and he is our manager who puts together these identification packets when they reach that final stage of preparation. He knows a lot about how things work in the lab, and he's also very happy to talk to you if you have questions about the science and how we do things in the laboratory. Now, with all that said, I want to yield all the rest of the time to one of our new colleagues, Ms. Leslie Chesson. Leslie Chesson is an expert, a world-renowned expert on stable isotope analysis. We recruited her, which you can read as we begged her to come work for us last summer because we were interested in adding a whole new line of testing to our repertoire in the lab. As you know, as you heard us say many times, we're always eager to add new technologies if it's going to make us better able to identify our fallen heroes. The newest technology for us that we're adding right now this summer is stable isotope analysis, and Leslie's going to give you a presentation on what that's all about. So thank you very much.
We'll see if I can actually advance the slides. Nope, there was one direction. Sorry, technical difficulties here. Aha! Thanks everyone for coming to this talk this afternoon. I'm very humbled to be here. As Dr. Byrd mentioned, I'll be talking to you about isotope testing, which is a new tool that we're developing in the lab. So I don't need to tell any of you this. This is something you know well, that the identification of commingled remains is very challenging. And you were thinking about, I come from a chemistry background, so this is something that the forensic anthropologists face every day in the cases of mass graves, things like plane crashes, and this is just an ossuary picture from Europe. So when you have these commingled remains, what tools do you have available to you to help separate and identify the individuals that are in the assemblage? Now, many of these you're, of course, familiar with because you've heard previous talks. These are techniques that I'm learning a lot about now that I work in a forensic anthropology lab. So, of course, sorry. Okay. We could be doing a little bit of this, I'm sorry. There we go. Can someone else advance these for me? Maybe that would be perfect. And one more. There we go. So these are the tools that are available now, of course, at the DPAA. And like I said, many of you have heard a lot about these. And you'll hear more about an update from the DNA side after this talk. But I'm here to talk about this new tool that we're adding, which is isotope analysis. So, can I get the next slide, please? Okay. Maybe. There we go. So what are isotopes? Isotopes are simply nature's recorders. They're a chemical tracking tool, basically. Anything that you eat and drink, you record in your tissues. And so you can envision them as a way to reconstruct someone's history based on the record that they contain within their tissues. So, what is an isotope exactly? It's simply a different form of a chemical element that differs in mass. So let's talk about the element carbon here for a second. So in most carbon atoms, we're going to talk a little bit of chemistry and physics here for a second. In most carbon that you find on the planet, you find 6 protons and 6 neutrons in the nucleus with 6 electrons orbiting. So this is how most carbon looks today. And the shorthand for this is 12 carbon, and you get that shorthand from adding up the number of protons and neutrons in the nucleus. Now, in some very rare cases, you get this guy, where you have an additional neutron in the nucleus. So it is still carbon. Functionally, it behaves the same way in chemical and biological processes, but it weighs just a little bit more because it has that additional neutron. And the shorthand for this, of course, is 13 carbon. These are both stable isotopes of carbon. They're just different forms. They're still carbon, but they're different isotope forms. Now, there's one other form of carbon that you've probably heard of. It's this guy, where you have 2 additional neutrons. So not surprisingly, the shorthand for this is 14C. And if you've heard of radiocarbon dating, this is the element that's used for carbon dating. It is not the element that we're looking at in the lab. So when we talk about isotope testing, we're looking at these 2 stable isotope forms, 12C versus 13C. So we have our isotopes. Another way to think about isotopes is like ice cream. So it's pretty hot out there today. I walked to lunch, was like, ooh, it's a little steamy. And I live in Hawaii now, so I'm saying that. But let's say you want to go and you're picking up some vanilla ice cream, and Breyers makes 2 forms of vanilla ice cream, a natural vanilla and a French vanilla. Now, what's the difference in between these 2? Well, if you look at the ingredients list, there's actually more stuff in the French vanilla. And I know that you probably can't see this on the screen, but I believe you have these slides in your packet. The French vanilla actually has just a few more calories per serving than the natural vanilla. So just like isotopes, if you were to eat all French vanilla versus natural vanilla, you might weigh a little bit more at the end of the day. So isotopes are like ice cream. Same thing functionally, but they weigh a little bit more in some forms. So how do we now use these? Next slide. Again, we're not using the radioactive kind, we're not doing any radioactive dating, we're only using the stable isotope forms in the lab. If anyone has read this book, The Omnivore's Dilemma, yeah, a few people. So there's a great quote in there by the author. When it comes to stable isotopes, he says, "We North Americans look like corn chips with legs." So here we are. Now what does that mean, corn chips with legs? We go back to the carbon isotopes. Carbon is found in carbon dioxide. So here it is, it's just out in the atmosphere right now. And if you're a plant and you're photosynthesizing, you have a choice, just like you have a choice in ice cream, about which flavor of carbon dioxide you're going to use. You can use 12 CO2 or 13 CO2. And depending on the type of plant you are, you have a preference in the type of carbon dioxide. So C3 plants, which are things like rice and wheat, preferentially use 12 carbon dioxide. Versus C4 plants, things like corn and sugarcane, which use more 13 carbon dioxide. So why do we really care about this when we're thinking about our corn chips and our diet? Well, if we go out and we measure all types of C3 and C4 plants, and this is a study that measured about 1,000 different types of plants, and they measured the carbon isotope values of those plants, you can see that isotopically, I can tell them apart. So those C3 plants, again, just to remind you, are things like wheat and most of your fruits, rice, C4 plants, the big ones are sugarcane and corn. Now, if you are eating C3 versus C4 plants, you go ahead and step through. Yeah, one more. You might look like a rice bowl with legs versus corn chips with legs. So I can now measure your tissues and predict something about what you are actually eating based on these carbon isotope records. And we eat a lot of corn in the United States. We produce a lot of corn, we feed it to animals, we use a lot of it as a sweetener in the form of high fructose corn syrup, so we have a lot of corn in our diet, just based on our dietary preferences versus other nations. So I want to talk about another isotope here. Next slide, please. So nitrogen isotopes reflect how much meat you eat, and it's basically a measure of the food chain that you're eating in. So if you're a vegan and you're eating no meat, you're only eating plant products, that's a very small food chain or a very small food pyramid. If you're eating corn or, chickens or cows, then they are just one step up in a food chain. So you're at the top. If you are now eating chicken, you're at the top of a slightly higher, longer food chain. If you're eating a lot of tuna, for example, in poke in Hawaii, we're at the top of a very long food chain because that tuna ate a lot of smaller fish, which ate a lot of much smaller fish. So the higher up in the food chain that you are eating, the higher your nitrogen isotope values. So I can say something about not only how much corn you might have been eating, but how much meat you might have been eating. With me so far? Okay. So you are what you eat isotopically. And what that looks like is something like this. And some of you may have seen this on the Facebook page that the DPAA has. So this is just, it's called them isotope biplots. We're looking at two isotopes. We're looking at carbon isotope values on the x-axis and nitrogen isotope values on the y-axis. And we can start separating diets based on these isotope values. So we have things which we might call a Western diet, where we have heavy meat consumption, lots of processed foods that have sugars in them, and you're going to have higher nitrogen and higher carbon isotope values, versus something that you might call a more Eastern dietary pattern. Rice, more plants, more vegetable proteins, less meat. So we can start saying something about your diet, again, just based on these isotope records. Then I want to talk about one more isotope, and this is an isotope that you find in water, so oxygen isotopes. So if you think about water, we have water. The biggest reservoir of water on the planet is the ocean, but that ocean water contains some heavy and some lighter isotopes of oxygen. Now, as you have evaporation over the water surface, over the ocean, the isotopes, the water isotopes that are lighter, actually evaporate a little bit more easily, right? Because they're lighter, it takes a little bit less energy. So you get water vapor that's isotopically lighter. That water vapor cools down, moves inland, you have a rain event. Whatever isotopes are sitting in that cloud that are the heavy isotopes, rain out first. So you get a rain event that has water that's isotopically heavier in the rain than its starting point, the ocean, but isotopically lighter than the actual, isotopically heavier than the rain, the cloud itself, and isotopically lighter than its starting point. So you see we're kind of getting this process of lighter and lighter values. And this just keeps occurring as the cloud moves inland and up elevational gradients. You guys are looking at me like, why do I care about this? Next slide, I promise it all comes. Yep, go ahead and show the map, sorry. Map, perfect. So, if we go out and collect buckets of rainwater, or we open our taps and measure the isotope ratios of water across the US today, the water isotopes vary. And you record that in your hair. Some of you may have picked up on this, but I lived in North Carolina originally. I have a little bit of an accent still. And then I moved to Utah right after college. So my drinking water changed isotopically between those two locations. And you could take a strand of my hair and measure the isotopes along the length of it and say, Leslie, I know you moved because I can see the isotopes that you recorded in your hair, they changed. And this can be really important. I got my start in looking at cold cases and reconstructing the history of an individual. Same thing for your tooth enamel. Where you lived and the water you were drinking as a child, you recorded in your tooth enamel. Okay, so that's how isotopes work. How do we now apply them? We're basically taking the records in a tissue, like bone or tooth enamel, and we're saying something, we're reconstructing something about that individual's history. So we can ask a series of questions. We can say, are these remains from a US American, for example? And we would answer that by looking at their diet and saying, well, we think that they were eating a lot of corn, a lot of meat, and that's part of a Western dietary pattern. If yes, then we would of course go on and analyze DNA. We can also look at your geolocation. So that's the idea of if we have information, where was an individual living during their childhood? If we're looking at that tooth enamel and we're reconstructing their history from that, we can see if it matches the home of record, which can provide an additional line of evidence for an identification. Make sense? Okay. So here's how we, the benefits. I should talk about why we're doing this. Basically, we're bringing, as Dr. Byrd mentioned, we are bringing this analysis in-house for the first time, and it's to help basically speed up potentially the identification process. Can we use it as a triage tool to separate remains and identify those that need to go on for further analysis faster? And can we help in cases where DNA evidence has not been conclusive because the remains were degraded and DNA analysis was not possible, there were no family reference samples available, so there are potentials there that isotopes can help in really tough cases. And then the commingling aspect, to bring this full circle, if we look at the isotope records that are in a set of commingled remains, can we group those that belong together based on these isotope measurements that we're making? So I want to talk, just to sort of wrap this up, about the K55, which you've heard a lot about, the 55 boxes that were returned a year ago today. And so we looked at the collagen, which is the protein that's found in bone, and we measured those dietary isotopes that I was talking about, this carbon and nitrogen isotopes. We tested 400 samples when it was all said and done, but we broke that sampling into two sort of phases. We did a pilot study, and then we went and finished the remaining 301 samples. And this just kind of gives you a timeline of how long it took us to do this. So again, they were just returned a year ago today. Sorry, next slide. Perfect. So here are the pilot study data. So what we did is we looked at the carbon and nitrogen isotope results, and we predicted whether we thought they may have come from someone eating that Western diet, so they were likely a US American, versus someone who was eating an Eastern diet, so they were a Northeast Asian, or what we determined was indeterminate. The isotope ratios didn't really tell us one way or the other definitively. So again, this is just the pilot study, so 99 samples, and you can see from that pilot study, about 63% of them looked like they came from US Americans. If you then go and look at the entire data set together, so all 400 samples, those percentages don't change a lot. So our pilot study was a pretty good representation of the entire assemblage, and a little bit, right around 60% or 57% look like they came from US Americans just based on the isotope data alone. And this is just looking again at that protein component in the bones themselves. So if we return to that biplot that I mentioned, if we look at the carbon and nitrogen isotope data together, this is how it breaks down. You can see the Western diet versus the Eastern diet. And I mentioned those indeterminate samples, the ones that we didn't think definitively fell one way or the other. And of course, those indeterminate samples and the Western diet samples are the ones that we would want to prioritize for DNA testing. And you'll hear more about that, of course, in the next presentation. So to sum everything up, here's the new tool that we're adding to the DPAA's toolbox. So the forensic anthropologist toolbox, looking at, can we use isotopes to discriminate based on diet as a triage tool to say you're more likely an American or not? Can we potentially look at where you're from? And this is that geolocation question, that origin question. Next bullet, please. Thank you. To compare the isotope record to the home of record we might have for an individual. And then, of course, to separate these commingling where DNA isn't possible in every case, or we don't need to potentially use DNA for every small bone, but we can reassemble or separate commingling using isotopes. With that, I believe that's my last slide, and I'll be, of course, around in the Q&A session to answer any questions. Thank you very much for your attention.
Okay, so next up is Dr. Tim McMahon, Director, DoD, DNA Operations, Armed Forces Medical Examiner System.
Next slide, please. So I'm the Director of DNA Operations for the Armed Forces Medical Examiner System. This is just an overview. I'm going to talk about who we are. I'm going to talk about the partnership we've had with the DPAA since 1992. And then I'm going to go through our capability capacity. I'll give you an update about the K55. And then we're going to talk about fit-for-use technology. Leslie introduced part of that is our unique development of things that are fit for use. There's a lot of DNA labs out there at your state, local level, and commercial, but not every method is fit for use. So I'm going to spend some time on that. Next slide, please. So who are we? We're the Armed Forces Medical Examiner System. We actually sit under the Defense Health Agency. We were established in 1990 as DoD's only human remains DNA testing laboratory, and we've been partnered with the DPA since 1992 to do human remains DNA testing. Next slide, please. We, just like the DPAA Lab, are an accredited forensics lab. We are accredited to the ISO 17025, which is the International Standards Organization for Forensics, and we are also accredited to the FBI Quality Assurance Standards. So what's that mean? That means we meet the same accreditation standards as your state and local crime labs, and so every 2 years we have people come into our laboratory to a new assessment, and every 4 years we have, for our lab, we had 13 scientists from 13 different laboratories come in, look at our processes and procedures, make sure we're following all of those standards. And last August was our reaccreditation, and we had 0 findings. Okay? Next slide, please. So we have a broad mission. I have 3 major divisions within me, underneath me. I have current day operations, or 12 scientists. They service the Armed Forces Medical Examiner System. So they do all the DNA testing for our current day operations. What came out of the past accounting mission was the use of family references in 1991 to ID a service member from Vietnam. Also in 1991, at the end of the Gulf War, there were a number of service members who could not be identified, and we had to use a commercial laboratory at the time. So it set up what we call the Repository Act. So right now, on file, I have 8.2 million DNA reference cards for all of our active duty, reserve, and National Guard. And because of that, 800 service members from Iraq and Afghanistan that would not have been identified by conventional methods were ID'd. So we have no unidentified people from Iraq and Afghanistan. But my largest group is my past accounting section. It's broken into 2 groups. There's the past accounting section, of which I have about 85 scientists dedicated solely to processing the samples from the DPAA laboratory. To assist them, I have a family reference section. There are about 12 scientists, and they're the ones who are collecting the references outside. They process those to create our family reference database. Year to date right now, of the original 8,100 missing from the Korean War, we have 92% covered with a family reference. That is what allows us to assist with identifications very, very fast. Next slide, please. So in 2019, back one slide, please. So in 2013, we had to build toward the NDA 2010, that capability and capacity. I had 6 teams at the time, and I had 48 scientists. Asking for additional money through a plan that we were supported by the DPAA. Between 2015 and 19, I was authorized 24 new hires. Next slide, please. Here we sit now. Instead of 6 teams, I'm at 8 teams. I'm at 90 scientists, and in fact, with that same budget of only being authorized 24 people, I actually was able to hire 58. Why? Because we got more successful and we brought on more efficient techniques. Next slide, please. So when we talk about DNA, there's two types of DNA. There's that really blue glob right there, that's called nuclear DNA. A lot of it came into the forefront with the O.J. Simpson trial. It's what your state and local crime labs use. There's a lot of misconception that nuclear DNA is the gold standard out there. Well, when I did criminal case, the question is who, against the general Zor population of 7.6 billion people, committed that crime? Nuclear DNA is definitely needed, that direct reference. But if we're dealing with a closed population, commingled set of remains, 14 airmen on a plane, you don't need to use nuclear DNA. You can use the second type of DNA called mitochondrial DNA. And those are the squiggly little organelles that you see right there. They give your body the energy to do. When we talk about mitochondrial DNA, that's the one that comes down only through the mother. When we talk about nuclear DNA, we actually have two tests that we can do there. We can do autosomal short tandem repeats. Those are the same ones your state and local crime labs use. Or we can test the father's side through the Y chromosome. Okay, so two types of DNA, three tests. Next slide, please. Our biggest problem when we deal with DNA that has been in the environment is degradation, okay, and other DNA, bacterial DNA. So when we extract a bone, not only do we get the human DNA, but we get whatever bacterial DNA is in there. So if I want to talk degradation, that's the chewing up of DNA. This is a very simplistic model. If I give you a 16-inch piece of string, and every year I told you to fold it, cut it in half, by year 6, you had 32 half-inch pieces. I wish it was that uniform. The way it occurs in nature, the best representation is if you go into your food pantry and you take that brand new box of spaghetti, you put it on the ground and you stomp on it twice and you dump it out, you're going to see all different sizes of spaghetti. That's what's occurring in nature. The longer pieces are the bacteria, the smaller pieces are the human. So we have to figure out how do we overcome that bacterial DNA as well. Okay? The basic unit of a DNA is a base pair. I just want you to think about when I say 1 inch, that's 100 base pairs. Next slide, please. So when we deal with mitochondrial DNA, we have the ability to test down to 1 inch pieces, okay, using specific primers that we developed or ways to hit those targets in 1998. Okay, so we can get degraded DNA from non-chemically treated samples. Next slide. Most labs will bring on one commercial kit for DNA testing of those nuclear DNA, that short tandem repeat. We've actually brought on all 4 kits. Why? Because sometimes certain kits work better than others. But if you look in this, that range on the top is 100 base pairs to 300 base pairs. So really, you have to have very large pieces of nuclear DNA. So we have figured out how to overcome this degraded DNA as well. And I'll talk more about that in a second. Next slide, please. When we talk about nuclear DNA, I want you to think of a tree, okay? So I sit at the base of the tree, the trunk is the thickest, right? I go up the tree, the trunk thins. That's because I know I have half the DNA of my parents, a quarter of my grandparents, an eighth of my great-grandparents. So I can measure that mathematically. When it comes to my brothers and sisters, that's like the branch, as thickest at the trunk, where I have 50% of the DNA with my brothers and sisters. It may not be the same 50%, but we have 50% in common. But as that branch goes out, it thins, and if you look at first cousins, you're down to 6.25% shared DNA. What's that mean? If we led with nuclear DNA, we would have to get 5 or 6 cousins to get the same mathematical weighted evidence of one YSTR. So we look to use a multitude of tests so that we can get the best results as we can. Next slide, please. This is a very busy slide, and it's a colored slide. Each one of those colors tell a different test that we do over the last 12 years, how they've increased. The really important part is that upper top part. I can tell you I do all these tests, and I can tell you I report out 3,000 analyses. But what if it was only successful 1% of the time? I'm not really a good lab. What this tells you is that for non-chemically treated samples, 94% of the time, I can get a mitochondrial DNA result. I can get a result for short tandem repeats, Y or autosomal, about 60% of the time. That's actually better than a lot of labs with current day samples. Now, next generation sequencing, that's the method we've talked about previously. That was developed for those chemically treated punchbowl samples. It took us 16 years to crack that code and to develop a method. And the reason for that is, remember I told you, currently we can do 1-inch pieces. Well, the DNA that we were getting out of those chemically treated were only a 1/2 inch. So we had to develop a whole new method that was not commercially available, fit for use. All right, next slide, please. How does that translate? Well, it translated into our product. Once we do a DNA and we get a result, we send it to the DPAA, we compare our database, we write what we call a believed to be report. And that believed to be report is the first time an unknown sequence hits to a family reference. Okay, so if you look in 2018 where there were 203 new identifications, we wrote 189 first time named reports. That means we supported 189 of those 203 were supported with DNA results. Okay, but if you look at the total number, it was 719 reports. Why? Because of addendums. We're always going to find additional portions in commingled remains, or if they go back to the site multiple times. And so when we look at 2019, we're already at 128 believed to be reports this year. So we're well on our track to supporting, again, 200 identifications. Next slide, please. So when we talk about the punchbowl sample, I talk about it as a mitogenome NGS. And why? Because if you think about a clock, the region between 11 and 1 is what we typically were testing for when we did mitochondrial DNA. This new method allows us to test the whole mitochondrial genome. And it allowed us to go after these chemically treated samples. In 2016, we became the first and only lab still in the United States that is accredited, that has this method online, as well as internationally. We're getting requests to support that internationally. So next slide, please. What this allows us to do is, we've all heard that traditionally there's a common mitochondrial DNA sequence. When we look in that region from 11 to 1, I'm one of those people. My mitochondrial sequence matches 7% of all Caucasians. But now we have the ability to sequence the whole mitochondrial genome. We're finishing up an National Institutes of Justice grant where we're looking at 1,000 US Caucasians, 1,000 Hispanics, 1,000 African Americans. And preliminary, we're looking, we can distinguish people with common sequences 98% of the time now. And so we can use this new method to help segregate those common sequences. Next slide, please. What this tells us with the NGS method is in 3 years, we've done 1,263 samples. 51% of those have reportable, but the biggest thing is it's led to 128 first-time named reports. That's 128 new identifications that we supported. And these are the chemically treated samples. So there was a question earlier about it. Did the K55 impact the Korea disinterment project? And the answer to that is no. The K55 are non-chemically treated, so they go down one pathway. The Korea disinterment are chemically treated, they go through this pathway. Next slide, please. So to support 200 identifications, this is just to show you that we've had to go from 1,400 analyses to 3,000 analyses, and I had to increase by 39 people. The director had mentioned that they're on a scale to increase scope and capability. Director McKeag supported us by sending a letter to our Director Vice Admiral Bono. We're on the same page. Next slide. We've asked for additional funding to increase from 3,000 analyses to 6,470 analyses by 2023, with the authorization to hire 32 new people. My boss at the Defense Health Agency, Vice Admiral Bono, is in complete support of that. We're looking to start this next year. So, next slide, please. So with the K55, we received 505 samples for testing. Okay, we had the initial samples were done by 8/23/18, and that was in support of the first couple of identifications. And then you can see that we had box 1 through 20 by mid-September and box 21 through 25 by October. In a nutshell, from October until 2 weeks ago, We have processed all 505 samples completely through mitochondrial DNA sequencing. So that is a full sixth of the analyses that we are doing in a year, we did in 9 months. We are now doing additional testing, nuclear DNA testing, which is allowing for the support of those 25 identifications and further on. And so we're really on track. And the reason was these were highly commingled. As you saw from Dr. Leslie's talk, that they were seeing about 21% of those were Asian sequences. Through DNA, it looks like we're seeing about the same percentage. And so we're really coinciding. We had to get through all of the samples so that we could segregate and make the identifications. Next slide, please. So why does DNA take so long? The biggest part is most of your instruments for our DNA testing are geared to criminal forensics. They're geared to these really nice samples, not geared to things that have been in the environment 70 plus years. The other part is a lot of times the method's not out there commercially, so we have to develop it, and it takes us time. And I'm going to talk to, next slide, I want to talk about fit for use again. And I'm going to give 2 modern examples that probably come to mind. Next slide. So everyone, any CSI fans here? All right, yeah, they do DNA in 45 minutes, right? Yeah. We're approaching that though. The instrument that you see here was developed for booking stations. And they can actually take a swab now and get a nuclear DNA result in 90 minutes. Okay? So these instruments, you may have heard about the California wildfires a year ago. Well, they were deployed out there to assist the medical examiner with doing DNA identifications. And they were robust. Next slide. However, there were some pros and cons to this. They were developed for swabs. They were developed for high copy DNA. And the cow bone you see in the lower left, you see all that red material here? That's really what they're kind of getting the DNA from, not the bone. So we have assessed it. We've worked with them. It's not going to work for the typical bones we see on the right-hand side, but 90 minutes, that's going to help us with our current day operations when we have DNA as the only mechanism to do an identification. Currently, I have to do it in 24 hours. It'd be great to cut it down to 90 minutes. So we are looking at that for a different avenue. Next slide, please. So what if you don't have a paternal relative, you don't have a maternal relative, or there's no brothers and sisters? We have a lot of people who are paternal nieces, right? Ancestry.com, 23andMe, we all heard of it, right? Okay. I want to take some time and talk about the Golden State Killer. We all heard about this last year, right? So next slide, please. So this individual, very bad individual, 1975 to 1986, murders, rape, burglary. What it boils down to is in 1987 or 1986, Sir Alec Jeffreys defined nuclear markers that could be used in forensics. 1987, it was used in England. The investigator of the last rape in 86 actually froze the criminal samples. Now we jump ahead, and they took this, they ran it against a commercial assay, and they used GEDmatch, and they were able to use his like Ancestry.com type results and track down a family and do investigative to identify the killer. Okay? So, next slide, please. We actually thought about this in 2015. We sent out a question saying to a company, we want, irregardless of your relationship, we want to use these nuclear markers that 23andMe and Ancestry.com are using. And irregardless of your relationship, as long as you're within 4 generations, we want to be able to do an identification. It was awarded to Parabon. We tried that commercial chip that the rest of the companies use. Yeah, it failed miserably. There are 800,000 markers. We recovered 25,000. Not going to work. So we had to develop a new assay. Next slide. They developed the assay in 2 years. We're on our 3rd year of a 4-year grant. In 2 years, they developed the assay and the software. We gave them 7 references and an unknown sequence with the testing method that they developed for us. We processed it, and it correctly identified the right reference with as little as 1,100 of the 15,000 markers. So we're looking to bring this on. We're finishing up the grant, and this would be another avenue for us to open up the pool of our results. But again, fit for use. Commercials didn't work, so we had to develop a new method. Next slide, please. So basically what we're looking to do is take a modern approach to DNA. We want to maximize the discrimination of the power of DNA. We want to enhance the laboratory process by using automation. We embrace that so we can free our people up to do what we need to do, which is the data analysis, so we can get the results to you, the families, faster. And then we want to use these single nucleotide polymorphisms and switch more to a nuclear-based testing method. Okay, next slide. However, we can't do this without you, the family members. So I told you, we have 92% coverage of family references. The one thing we're looking is in 2013, we really started testing short tandem repeats. So if you have concerns or questions, stop by our desk. We can tell you who or what type of references we have already. We've been very successful collecting, so everyone who's donated, I want to thank you. But you, the family members, are the biggest asset that I have to making identifications. The biggest point I want to point out on this slide is it is a medical record. We protect it by HIPAA, just like you go to the doctor and signed it. So if we tell you we have an aunt and you ask us the name, unfortunately, I can't give you the name. Okay. So next slide, please. I know I covered a lot of stuff very quickly. At both our website and the DPAA's website, there is a common DNA questions there. There are about 27 questions that go very into detail of everything that we do in DNA testing, and my contact information is there as well. Do not hesitate to reach out to me. Next slide. With that, I thank you very much for your attention. Thank you.
Okay, so folks, next up will be our Q&A session, but before we do, just since we started here about an hour ago, we now have 3,200 views on Facebook Live and over 4,000 people that we've reached. And also, I was supposed to do this earlier, and I apologize. Mike Baziak, am I pronouncing that correctly? So Mike, if you could go ahead and stand up, please. Mike is a point of contact for the Revisit Korea program for families of the Korean War MIA KIA servicemen. So if you have any questions about the program, please get with Mike as a point of contact. Okay, so I think what Mike said is that she does a better job than you. Okay, I thought that's what I heard. Okay, but thank you. So if you have any questions, please, you have two great points of contact that you can reach out to. Okay, so again, rules of engagement before we start the Q&A session is raise your hand, a microphone will come to you, and we would ask that your questions be directed towards the briefings that we just received. So with that, I'll open it up to the Q&A session.
Thank you for your briefing. My brother was MIA in July of 1953, and I was in college when my father notified me that he was missing. And he had written to me before he went missing, and I came to one of these meetings and was asked, I mentioned that to someone, and they asked me to send them an envelope because back in those days you had to lick it. You remember? Maybe you're not as old as I am, but believe me, you used to have to lick your envelopes to seal them. And I sent that one of the envelopes off to them, and several months later they sent it back to me, which I thought was very nice, but there was nothing in the correspondence about how useful that had been. And I'm curious about how useful would that be?
So, afterwards we can get together and talk about your specific case. But in reality, envelopes work about 1% of the time. And a lot of times they come up with a mixture because people, even though they licked them, people have touched them and things like that. We use them as a last resort when we do not have any other form of a reference out there. We do that. But in fact, better than references, we will talk with families that don't have viable references. A lot of times there were baby books that were done at the time. First hair cutting will give us mitochondrial DNA. Baby teeth work very well. The baby teeth are really small, so we end up consuming the whole tooth. But, and then there are the old style watch bands. My grandfather had one where the watch band expands and there's the metal. It traps a lot of skin cells in there and it works really well. So we do have a lot of alternative references in case we don't have a viable reference. But that was an excellent question.
Okay, so just for clarity here, I was told earlier that I didn't call on the back enough, but I'm seeing only hands in the front here.
Thank you very much. This was fascinating. I have a question about the isotope study. While I love the Frito reference, the American diet has changed dramatically since World War II. And if I'm looking, like, for example, at our family member and the socioeconomic, cultural background which he grew up, which would have dictated his diet, it wouldn't have included Fritos. I know that's just an analogy. But can you speak to how you address that or how you take that into consideration when you're looking at diets? Thank you.
It's a very good question. Thank you. So there are, we can get an idea of what the population looks like by looking at published records like skeletal collections. So folks who we know their birth and death dates, and then what the isotope ratios or the isotope records in their tissues look like. And so we can get some contemporary reference populations. So we would know, well, this is what the American diet isotopically looked like recorded in a tissue in 1950 or 1960 or today. And even though our diets are changing, as Americans we still eat a lot of corn compared to Europeans or Southeast Asians and Northeast Asians, and so these patterns are generally holding true, but it is an area of research in the isotope community at large. Does that answer your question? Thank you.
My name is Leo Linda Bowers, and my uncle was MIA in Korea in December of 1950. We are from the big island of Hawaii, and there are many people here from Hawaii. So how do you account for the high fish intake?
That is an excellent question. I will say that we don't know yet, but it is something that we are looking at. We do know in general what populations look like, but there are some what we could call sort of distinct dietary patterns. Another one that we see a lot are natives of Alaska. They have enormous marine inputs into their diet, and isotopically they are very distinctive. So again, I'm sort of speaking for the entire isotope community, but it is an area that folks are researching and are looking into. So thank you for that question.
I will say that Spam does have a signature like the mainland. we eat a lot of Spam in Hawaii. So.
I don't know if I can top that, but my question is, so you have commingled remains, and I don't know how to put it, but so you're going through all of them and you go bone by bone. Do you wait till, what if you get like, do you wait to go through all the bones before you notify a family, or how does that work? You following me there?
I think I do.
Okay.
And I will tell you that for the K55, Dr. Keys is going to give kind of an overview of the process. And last year here, I told you what we were going to do, right? Projecting ahead and how I thought it was going to work out. Dr. Keys is going to look back and tell you what we did and how it's working out now. I will tell you the predictions were pretty much on, but without reproducing what she's going to tell you tomorrow, just in short, I'll tell you that we went through all of the boxes, and you may recall at this time last year we had 2 identifications that we made, and it was Master Sergeant McDaniel and Private Jones. Those 2 we could make quickly because we could see things that told us first off told us we ought to be able to identify them quickly, and then when we did the work, we were able to identify them quickly. And even the DNA lab turned the samples in a matter of 2 to 3 weeks, and all the other reports got done quickly. The rest of them weren't that easy, you know, and we were, you know, kind of warning you last year that some of these are going to be harder than others. And but as you'll find out tomorrow when Veronica goes through the process, there were many different steps involved and a lot of different people involved in this process.
And from a DNA standpoint, when we get the commingled remains, once you start getting above the 60% processing of those samples, you have a really good map at that point of the spectrum of what is common sequence and what is a non-common sequence in mitochondrial DNA. And it allows us to start to segregating. When I tell people it's like a jigsaw puzzle, if I gave you a 5,000 piece jigsaw puzzle with none of the border pieces, you'd probably want to kill me because you'd have no way to start. What we did with that step is the first round of getting through the mitochondrial DNA is establishing those borders, and then you get those pieces where that little tab is just a fraction larger than the one next to it, and you're trying to figure out. That's what the additional testing or the clavicle or the isotope testing can help with. One of the things in support of the isotope testing is with the terawa samples that we receive from the atoll that are being recovered by history flight. About 40% of them come to us as Asian. With the isotope testing, if they can keep those and give us that, it's going to speed up our identification process. So it's this community effort that we have between the labs and developing these new techniques that's going to speed it up over time.
Hi, my question is regarding nuclear DNA shared between relatives. Our family member who was killed in Korea, the DNA that was given by his sisters, which was same mother, different father. But we just realized that we have his mother's hair. She cut her hair in 1921, we still have it. I wonder if it would be advantageous for us to submit that to you.
No, so unless there's the root that's on it, so when you look at your hair in your hair brushes and stuff, if you see a white portion at the bottom of the hair, that's what we call the follicular tag. That's where the nuclear DNA is.
But you just said earlier that if there was hair from a baby book.
For mitochondrial DNA.
I see. Okay,
So
thank you.
Yep, good question.
Hi, I have a kind of a technical DNA question. I'm wondering if you can identify what statistical software packages you use to match the nuclear DNA and mitochondrial DNA. Do you?
Yeah, so we currently use one called DNA View, but we're in the process of validating an in-house one that we're calling LSAM, Lisa Statistical Analysis Method. We've built it with statistical assistance to be more robust, but basically what the software allows us to do is for nuclear DNA, we can take the unknown nuclear profile, and then we can take the references and say, okay, this reference is the sister and this is the brother, and we can do what we call a sibling statistic on it. Or if it's a mother and father, we can do a paternity statistic on it. And so what it allows us to do is get that nuclear statistic if we do it, but then we have the mitochondrial, and then we have the Y. If we get all three results, we can actually multiply those together, and we issue what's called a likelihood, which states it's X many times more likely that this reference belongs to the biological sibling of reference so-and-so type thing. But yes, we use a DNA View currently, and then we're developing more robust one in-house.
So we have a question all the way in the back.
Hey, we've got a question from our Facebook feed, and before I get to it, I just want to say thank you to those of you in the room who have been checking in on the page. I see your comments, and I appreciate you checking in and chiming in on the feed, even though you're still here in the room. That's great. So Anthony on Facebook asked, and this may be a little more appropriate for some of our morning session, with a large number of DNA samples on file, will the Korean War unknown from the Tomb of the Unknown be exhumed at any point?
the Tomb of the Unknown in Arlington Cemeteries is not included in the disinterment plan that we're currently pursuing. All of those disinterments are in the Punchbowl. But you may recall we were asked the question last year, and what we said last year was that once we get through the entire 7 phases of the current disinterment plan, then we could maybe take a look at that. But of course, the permissions for that would be a long process in and of itself. But from a forensic standpoint, there's no reason why we wouldn't at least give it a look. But it's a ways out.
Hello, my name is Ann, and my dad is missing. And as a family member, I was wondering, how do I know that you have enough of the right kind of samples from different people in my family so that the mitochondrial and nuclear DNA matches could be possible?
So that's an excellent question. For every meeting like this, the Korea Cold War meeting, or for a general family member update, the DPAA and the Service Casualty Office provide us with a list of attendees and who's attending, and then we will take that and we will compare it to our family reference database of what we have on file. So if you stop by, see me afterwards, or stop by my people outside, we can actually look up your loved one and tell you what we have on file.
This question probably relates to what you just addressed. If somebody gave a DNA sample about 10 years ago, and I understand that was for the mitochondrial, is it necessary to go back so you have information for nuclear testing?
So if the person who, let's say it's a brother, sister, mother, father, gave it for mitochondrial DNA, we have that reference on file. It automatically rolls into the nuclear DNA testing.
I had a DNA question and an isotope. The DNA, we were able to submit a blood sample from the brother of our service member, but there was something about making sure it was registered to be released or used. How do we make sure that you have permission to use it?
So if the blood sample came to us, typically, so we collected blood references up until about 2003. Mm-hmm. They were collected under an informed consent where a phlebotomist actually came. You can stop by again, see me, or stop by and see my people at the desk. If we had received that sample and there wasn't the informed consent, the service casualty would have reached back out to you.
Okay, thank you.
Yep.
And in the isotope, you had talked about how your hair would indicate your changes in location. So in these older bones, does the isotope stay from their time when they were stateside?
Very good question. So basically, we remodel different tissues at different rates, and some we don't remodel at all. So our hair and our fingernails were continuously growing, of course, so those record the last weeks and months of our life. Our tooth enamel, we form it when we're a child or young adult, depending on the tooth, and then it doesn't change. So those isotope records that you put down, that you stored at that point in your life, you still have. And so those can be very useful. You basically are getting different snapshots in time. And then your bone, we slowly remodel it. So depending on the bone, it might represent 5, 10, 15 years of your history. So again, if we can get different tissues, we can tell different, we can look at different points in your life. There's different snapshots in time. Really good question. Thank you.
I have a question on the 55 boxes. Do you have an idea from what you've done peripherally to begin with, what part of Korea they're from? What area? Are they from the Chosin Reservoir? Are they from the West? Are they from the demilitarized zone? Do you have any idea what you're thinking?
We do. When they came in groups of boxes that had a village name with them. By far the largest group comes from East Chosin, and in fact that was the last of the DNA samples that they have finished up this summer. There was a lot of East Chosin samples, and we're making a lot of identifications from East Chosin now and in the near future. Last year, you may recall we made ID from Unsan and Kujang. That would be from Battle of the Chongchan. But every box has a village name with it, and every village name we can associate to a battle that took place. And so most of them are East Chosin. The second largest group would be Unsan Kujang area.
And when we talk about the DNA of those 55, remember I told you we had a 94% success rate, and that's really with any sample from Korea, Vietnam, World War II. These actually 55 boxes, our success rates for mitochondrial DNA are 98%. So they're actually working really, really well, 60% again for STRs. And so they've been really good samples to work with.
So I had a question regarding to x-rays and medical work. Our uncle had an issue where he had a thumb growing off of another thumb on both hands, and it was removed before he went into the Army. If you found remains like that, would you be able to find that, would that evidence of that still be there to help identify?
It's kind of hit and miss with the medical records for the Korean War. We do have a small percentage, something on the order of 5 or 6% of our Korean War missing. We have some medical radiographs in the file, but we don't have medical radiographs for most of our Korean War missing. So in your case, if you talk to your service casualty officer, ask them to ask us, we'll look it up. And we'll see. Ma'am? Oh, okay, yeah. It could help us potentially. It depends on what we have in the lab to compare to. Sometimes they're extremely helpful, and then other times, if we were missing, say, if we were missing an arm and it pertained to the arm, it couldn't help us in a case like that.
My name is Marina Oros. My brother was Joseph Piver Jr., Corporal. He was killed in Korea. There were doubts about how he died, whether he was a heavy weapons crewman, and it could have been blown up by a mortar shell, someone witnessed. And I was wondering how small, or could they find anything of him to do DNA on?
So that's actually an excellent question. So up until 2006, we had to deal with larger fragments of bone from the DPA on the average of about 5 grams, which is about 1.5 inches piece of bone or so. In 2006, we developed a new buffer that actually pretty much every lab in the world uses now. We call it the demineralization buffer, and it's basically dissolves the bone completely. And now we can test a bone about the tip of your pinky. And so there is, if it was blown up and they find small fragments of bone, we've tested extremely small pieces of bone to the tune of about 0.1 gram of bone. So if you have a food scale, you know what 0.1 gram is. It's like one piece of dry spaghetti. So we actually can test a lot smaller than we have, and we're pushing that limit now even further.
And then to make matters worse, they further examined with some of his fellow soldiers, and they said they saw him in a ditch at night when the Chinese were coming down, and that he was there, and they don't know what happened to him. So I don't know whether that's what happened or not. And there was another thing we asked in the very, very beginning, so far back when it happened, that they said when the people who were buried in Hawaii, you couldn't get DNA because they embalmed them so much. So have they taken care of that now?
Yes, so part of my talk was when we talked about next generation sequencing, or that NGS method. We worked with the DPA. It took us 16 years, but in 2016 we actually developed a method to get DNA from those chemically treated, and is which is part of the support of the whole Korean Punchbowl disinterment plan. Is we have a mechanism for testing that. So we can test it chemically treated, we can test very small samples. One thing I think we're remiss in saying enough of is as our partner with the DPA, we talk daily and weekly. If we get a sample that we can't get a DNA result from, in clavicle or something, we don't forget about that sample. We're all looking to develop new methods, and every time we bring a new method on, about every 2 years we're bringing something new on, we sit and we review those cases. And if they're fit for use at that time, we will start retesting them. We don't give up, is the bottom line on this.
You sound like my brother. He didn't want to give up either when they were coming at them, and they stayed, and they gave him the Medal of Honor. And the whole unit got it from standing on and not giving up. They wouldn't give up, so they died for that.
Well, thank you, ma'am. That's the highest praise I think I could ever get.
God bless you for doing that. Thank you very much.
Hi, simple science question regarding isotopes, or maybe not so simple. You mentioned that you can tell based on the diet, whether it's a U.S. American, Western diet, or perhaps a not U.S. American based on the Eastern diet that you find. What if someone grew up as a U.S. American but did not grow up eating a Western-based diet? I'm presuming that isn't discounted, that you continue to do testing perhaps?
Absolutely. So we talk about isotope testing being just another tool in the toolbox. And just like many of the identifications that come out of the lab use multiple lines of evidence, this would be an additional line of evidence. And it may not be the best tool for every case, whereas there are other, of course, tools available, things like DNA. So we just want to give another tool that can help support an identification, potentially exclude a possibility, but it's not the sole tool that's available.
You stated it was the 55 boxes that some of them were designated with villages up around Eastern Chosin. I was wondering if any of those boxes were marked from UDAM-NI.
No, sir. I think the marking was Chansong-Ni. Excuse me? I'm sorry, yeah, Sinhong-Ni, East Chosin, yeah.
Hello, my name is Wes. My great uncle was from Texas. He's a bright red area on your geolocation map. I had a question that was a follow-up on a previous answer about drift of the isotope ratio over time, depending upon time in Europe plus time in Korea. If that is I know it depends upon the sample that you have.
Absolutely.
And I had another question about why DNA.
Okay. So yes, sir, absolutely. It will depend on the tissue that's available and what time period in an individual's life we think that tissue represents. So again, tooth enamel is a great one for childhood, young adulthood, and that's why these home of records become so important for us, so that we can match to something like that bright red spot in Texas, that bullseye in Texas is what we call it.
And you didn't discuss why DNA too much. Is it not useful?
No, it's absolutely. So
does it survive?
It does.
It does survive?
It does. So when we talk about the two types of DNA, there's nuclear DNA, and that's a single copy. There's mitochondrial DNA, and there's anywhere from 10 to 100 copies of the mitochondrial per cell. And so over time, initially, it was easy to get mitochondrial in the 90s because there was just so much of it, and they were in that single copy. But when you look at your nuclear DNA, there are 23 pairs of chromosomes. 22 of those are what we call autosomes. They contain the markers that your state, local, and federal crime labs use. We use those as well. They're the ones that track what we were talking about, that paternity, how we match you to your mom and dad or your brothers and sisters. The 23rd pair of chromosomes determines if you're male or female, and that's where the Y DNA is. And in fact, in the order of testing, we have the service casualty look for maternal references, followed by YSTR references, because it's easier to get those. And we have, when you look at our success rates on that one slide, It's about 67% for Y STRs and about 60% for autosomal STRs. So we use it in abundance. It's an excellent testing. It survives. We actually just had to modify the actual procedure to fit these samples. And so we had to augment it and make it what we call a low copy procedure. And a lot of labs don't use that because they're afraid of what we call contamination. And so our lab, we track our contamination, we will bleach down and we control for it, and it becomes a very strong method to support the identification process. So excellent question.
We've got another question from social media, and again, for those that are viewing on any of our platforms, please put your question in the comments. The question is, can you get isotopes from bone you can't get DNA from?
Very good question. We believe so, but this tool is one that we're developing right now. It will go up for accreditation this year. So this is literally the newest tool at the lab, and we'll do lots of validation studies. And so one of those will, of course, be what happens with some of these difficult samples, these things that have been embalmed, that DNA testing was tough to do. Would isotope testing potentially help in those cases? So it's an area of active investigation. So stay tuned.
All right, folks. So that'll wrap up this portion of the Q&A. If I can get a round of applause for our briefers. Okay, so a couple of quick announcements before you're released. And the first one is, do you like the way I said that? So y'all are paying attention. I like that too. If you are attending tonight's dinner, which starts at 7 p.m. right in here, and you have not picked up your ticket, please pick up your ticket. In the back forum area, they have a ticket counter. The MPVA has a ticket counter. Pick it up by 4. Or they will start giving those tickets out to the folks who would like to attend. And then as a reminder, I need everyone to take all of your personal items tonight because this room will be completely reconfigured. And there will be no assigned seating tomorrow. There still will be the table numbers, but no assigned seating. So when you come in tomorrow, if you want your same table, maybe get here early, but just understand that there will be no assigned seating for tomorrow. So with that, thank you, everybody, and look forward to seeing you tonight at 7.